All right, so we are back again, and now we continue from where we left off. So where did we leave our last class at? We left at the brink of an important metabolic process in the metabolic, or rather general metabolism of amino acids. We were studying that, and we just left at the brink of an important cycle, which is the urea cycle, right? So the urea cycle. Now the thing is, why and what is urea cycle? Why is this needed at all? And firstly, what is this thing, urea cycle? So just for bringing back the reminiscence from the last classes, we said that ammonia is an important product which is formed from the metabolism of amino acids. Why? Because amino acids have this defining group known as amino group, which contains nitrogen, right? So the nitrogen part of amino acids, that is removed by two consecutive metabolic steps, that is transamination and then oxidative deamination, right? After these two steps, what we essentially get, apart from an array of other products, is a very important metabolic waste product in our body known as ammonia, right? Now there is a problem with ammonia. Ammonia is highly toxic in our body. It is so toxic that even the minutest increase in its levels in the bloodstream will lead to very dangerous consequences. All right, so we don't want that. Our body obviously does not want that to happen. So what our body does is remove ammonia. Now there is another problem in that. Our body cannot remove ammonia as such. It cannot remove ammonia in its own true form. So what our body does is to improvise with the situation is that ammonia, which is very highly toxic, is converted into this compound urea. This urea is quite less toxic, and it is metabolically inert also. Also, this urea is highly soluble in water. So large amounts of this urea can actually be excreted out. So the conversion of ammonia to urea is what constitutes our discussion today in this video, that is urea cycle. This process is taking place. So now obviously when we are talking about a process, a series of steps is happening here, and so we need to understand what are the steps that are happening here, right? So first we start with our topic of discussion, that is ammonia, right? So ammonia, we know, is present. Along with ammonia, for urea cycle, we need another molecule, another compound. What is that? Another starting compound is very important, carbon dioxide. So ammonia and carbon dioxide will react in the presence of an enzyme. All right? In the presence of an enzyme to form a compound known as carbamoyl phosphate. All right, carbamoyl phosphate. Very important. The enzyme needed in this very step is carbamoyl phosphate synthetase, right? Carbamoyl phosphate synthetase. So that is exactly the enzyme which is needed. Carbamoyl phosphate synthetase. Quite easy, right? This is the enzyme which is needed. Anything else? Yes, another thing. This very step is actually energy demanding step. So ATP will be required, right? ATP, ADP plus inorganic phosphate, right? Now do me a favor and remember not only the fact that ATP is required in this step, but also the fact that 2 ATP are required. Can you do me this favor and remember this? Yes, 2 ATPs are required in this very step, which form 2 ADP plus 2 inorganic phosphate, and the energy of this very ATP breaking is required in the reaction ammonia plus carbon dioxide, giving carbamoyl phosphate in the presence of the enzyme carbamoyl phosphate synthetase, right? Now this step has another very important feature. What is that? This step is the rate limiting step of urea cycle. Very important. Very important. I cannot stress enough on the importance of this step. That is the rate limiting. Alright? Alright. Now let's proceed to see what else is there in view for us. Now the carbamoyl phosphate which is formed reacts with a compound which is ornithine to form citrulline. Alright, great. The enzyme which is required in this step is ornithine transcarbamylase. Alright, so let me write that up. I'll be explaining this obviously. Ornithine transcarbamylase. Alright, ornithine transcarbamylase. Now, listen to me very carefully. What is happening here? There are a couple of points which I need to cover here. When I said about the starting of urea cycle, since we have started already urea cycle, we forgot to mention something. Can you say something? Can you say what I forgot to mention? Yes, the site of urea cycle. The site of urea cycle is a bit, we can say, interesting. Urea cycle is what we say to be compartmentalized. Yes, it is compartmentalized, which means that some part of urea cycle is happening somewhere in one compartment, some other part is happening in some other compartment. So it is well compartmentalized. All right, which part is happening where? We have obviously not completed the cycle, but the steps which we have covered till now are happening in the mitochondria. Very important. Please note this. All the steps which we have dealt with till now, we have dealt with 2 steps. That is ammonia plus carbon dioxide in the presence of carbamoyl phosphate synthetase is giving carbamoyl phosphate, which is reacting with ornithine in the presence of the enzyme ornithine transcarbamylase to give citrulline. Up to this, everything is happening in the mitochondria. Alright. After this, citrulline will leave mitochondria and will enter the cytoplasm. Very similarly, ornithine had entered mitochondria from cytoplasm. So am I clear? This reaction is taking place in the mitochondria. Strangely enough, ornithine is not mitochondrial. It is not mitochondrially located. It is located in the cytoplasm, the cytosol. So it actually migrates from the cytosol to the mitochondria, right? Why? So that this reaction can take place in the mitochondria. Similarly, citrulline, after being formed in the mitochondria, migrates to the cytosol. Why? Because the rest of the steps of the urea cycle will take place in the cytosol. Right? Right. Now, citrulline, after being formed, will form with the help of another compound, which is aspartate. This citrulline and aspartate will react and they will give Argino Succinate. They will give Argino Succinate, right? Now this step is again very important. We had dealt with 2 ATP being converted to 2 ADP, hence energy being utilized in the first step. The third step is the next energy requiring step. Alright. So ATP will again be utilized here. Strangely enough, what would be formed? ATP to ADP plus inorganic phosphate. But in this case, there will be breakage of 2 phosphate bonds. So 2 inorganic phosphates and adenosine monophosphate are formed. So there are breakage of 2 energy bonds here and the breakage of 2 energy bonds here also. Difference is there are 2 ATP molecules here forming 2 ADP plus PI, and here 1 ATP molecule directly forming 1 AMP plus 2 inorganic phosphate. Right? Right. So 4 high energy bonds are required till now. And we will be seeing in the urea cycle there are no more energy demanding steps. So with these 2 steps, we complete the energetics of urea cycle. Although there is a little bit more, I will be talking about that later. Now do hear me out. What is the enzyme here? What is the enzyme here? Argino Succinate. What would I say? Yes, synthetase. See, whenever I do this, Argino Succinate or carbamoyl phosphate, synthetase. Argino Succinate, synthetase. Here, this is ornithine transcarbamylase. This is known as transcarbamylase because there is a transfer of the carbamoyl group from carbamoyl phosphate to ornithine. See, again, we are delving into the molecular part, the entire organic chemistry which is behind this entire biochemical process of urea cycle. So I feel like that would be too much as far as our course or our intentions or our focus is concerned. So we are going to skip the molecular part here, obviously, as I have explained before. But the name ornithine transcarbamylase, we can obviously remember that, knowing the fact that this enzyme is assisting in the transfer of a carbamoyl group from carbamoyl phosphate to ornithine, forming citrulline. Good. That's easy to understand. Now do me one favor and remember one more thing. There is removal of a molecule of water here. Alright, there is a removal of a molecule of water here. Right. Now, just remember this, alright? Now, what was the name of the enzyme? I didn't say that. That is Argino Succinate. I am abbreviating Argino Succinate Synthetase. Argino Succinate Synthetase, right? Now, this Argino Succinate, after being formed, breaks down. Breaks down means there is lysis of Argino Succinate into what? Argino. So the lysed product is arginine. Alright, arginine. So one lysed product is arginine, the other lysed product is what? Yes, very, very important, fumarate. So arginosuccinate lyses, breaks down into arginine and fumarate. All right. So what can be the name of the enzyme which is needed here? Very easy. I said lysis. So Argino succinate lyase. Argino succinate, abbreviating lyase. That is the enzyme which is leading to the lysis of this Argino succinate into arginine and fumarate. Now listen to me very carefully. When I say fumarate, does something come to your mind? Think. Yes, you've reached it. Yes. Fumarate, as we have read for a very long time, is one of the products of Krebs cycle. Yes. Krebs cycle, one of the intermediates was fumarate, right? So from fumarate, if we continue Krebs cycle here, suppose the fumarate which is formed here goes on to Krebs cycle. So from fumarate we will get what? Malate. What was the enzyme needed? Fumarase. Fumarase, right? From malate, what would we get? Malate dehydrogenase, oxaloacetic acid. Malate dehydrogenase. This is the part of the Krebs cycle, all right? We are saying that the fumarate which is formed after arginosuccinate lyses, the fumarate goes into Krebs cycle. So from fumarate, we would be getting malate to oxaloacetate. Is there a link between oxaloacetate and aspartate? Yes. The transaminated product. Yes, previous class. The transaminated product of oxaloacetic acid is none other than, yes, my friend, aspartate. Very good. So we have successfully linked Krebs cycle with the, for now, apparently incomplete urea cycle. We will complete this. So Krebs cycle and urea cycle are linked together. All right. So after we complete this urea cycle, there is just one more step left. After we complete the urea cycle, we will be saying that these two together, since they are both cycles, so it's as if we say that to be a urea bicycle. All right, since both of these are cycles, so urea bicycle is this. All right. So oxaloacetate to aspartate, we already know the enzyme is AST, aspartate transaminase, or aspartate aminotransferase rather, or as I said, GOT. I had said this in the previous video. That is glutamate oxaloacetate transaminase. Right. What was the site of Krebs cycle, by the way? Mitochondria. So you see, again there is a compartmentalization. This is happening in the cytosol. After I finish this will be happening in the cytosol. This part is happening in mitochondria. This part is also happening in mitochondria. This part is also happening in mitochondria. All right, let's complete it, and then we will kind of fill in the gaps. So arginine, after being formed, this is arginosuccinate lyase, right? Arginine after being formed, this is the last step, that is the enzyme arginase is needed. A molecule of water is converted into urea. This is the entire thing. All right, what happened here? Arginine had already been formed, right? From arginine, we get back ornithine, hence completing the cycle. What is the secret? The secret is this enzyme arginase. Also, there is another secret, that is H2O, a molecule of water. A molecule of water is needed in this, and the arginine apparently splits again into ornithine and urea. So this urea being formed from here is why the name of the cycle is urea. Oh, sorry, it had gone out of the camera frame. The urea cycle. All right. So this urea is being formed from where? Ammonia. So ammonia and carbon dioxide were what? The starting products, the starting material, and the urea is being formed. What is the molecular structure of urea, by the way? Very easy. CO NH2 NH2. Why I stressed upon the molecular formula of urea is because of these two things. What are these two things? Two nitrogens. There is a common question, that is, what are the two sources of the nitrogen of urea? Apparently, the two nitrogen of urea are derived from two sources. One is obviously directly from the ammonia, right? The other is from where? Yes, this aspartate. Aspartate is an amino acid, right? Right. So aspartate is the other source of the nitrogen of urea. Two nitrogen, two different sources. One is ammonia, one is aspartate. Right? All right. So with this we complete at least the overview of the steps of urea cycle. Now any metabolic process, it would have some, inhibitors and some promoters and stuff like that. So we will be talking about that and a little bit details of the steps as well. So if we have to start, let's start with the first step itself, right? So this is the filling the gaps part which I was talking about, right? We are talking about the information which we might have missed in the entire cycle, because this is a big topic, urea cycle. So the thing is, ammonia plus carbon dioxide is giving carbamoyl phosphate. This is a very, very important step because this is a rate limiting step. I talked about this enzyme carbamoyl phosphate synthetase, CPS. All right, carbamoyl phosphate synthetase is abbreviated as CPS. Now there is a very interesting thing about this CPS. This CPS has two isozymes. It has two forms. There is CPS1 and CPS2. So there are two isoenzymes, right? The one which we are using here is CPS1. All right, the one which we are using here is CPS1. By the way, let me actually mark the parts of mitochondria because that would be required. All right, so carbamoyl phosphate up to citrulline, kind of, because citrulline goes out, so I'm ending the border here to ornithine. This part is the mitochondria part. All right, again there is a mitochondria part here, that is fumarate. Fumarate, after being formed into the cytosol, again goes into the mitochondria because fumarase enzyme is located in the mitochondrial matrix. So fumarase, so this part is again here, oxaloacetic acid. Up to here, this is Krebs cycle. After this, this is transamination. So this is again cytosolic, not mitochondrial. So this part and this part are mitochondrial. Rest of the parts are cytosolic. All right, I hope I made myself clear. Now what I was talking about was the CPS1 and CPS2. You see, CPS1 is the enzyme which we used here. This is mitochondrial, but CPS2 is cytosolic, and CPS2 will be useful to us when we will be talking about nucleotide metabolism, especially pyrimidine metabolism. Pyrimidine metabolism, all right, that is where CPS2 will be used, right? Now I was talking about effectors, right? Positive effector, negative effectors of the different steps of urea cycle. This first step is very important because I'm perhaps telling this for the 1000th time that this is a rate limiting step. Now, there is an important compound which positively affects carbamoyl phosphate synthetase 1, and that compound is NAG, right? Which is N-acetyl glutamate. This is a very important positive effector of carbamoyl phosphate synthetase 1. Alright, NAG, N-acetyl glutamate, is a very important positive effector of this CPS1. Alright, why it is very important, this will be evident to us when we will be talking about the disorders related to urea cycle, which we will be talking in a few minutes. Alright, so NAG, which is N-acetyl glutamate, how is this formed? Be just a single step reaction. I think it would be better if we actually remember it. So that is why I'm telling this. That is N-acetyl glutamate. How it is formed? Glutamate. It is there. Glutamate. From glutamate, we get N-acetyl glutamate. So glutamate, done. What is this? Acetyl. So we need an acetyl something to donate the acetyl group. What can be the donor of acetyl group? Very good. Acetyl CoA. Acetyl CoA. So acetyl CoA, and it becomes coenzyme A because it gives on the acetyl group. And obviously this is enzyme dependent. So the enzyme which will be required is N-acetyl glutamate. Very good. Synthetase. Now this has become kind of a habit. NAG synthetase. Very good. That is the enzyme which is required here. Alright. Now if we are to break down this N-acetyl glutamate back to glutamate, we would require the enzyme decyclase, or rather, I'm sorry, deacylase. What the hell am I talking about? Deacylase. Deacylase, right? Because you are actually deacetylating N-acetyl glutamate. So deacylase is the enzyme which is required. And in that case, what would be the acetate would be given out? And also deacylase is, this reaction is actually kind of a hydrolysis. So water is also required. Alright. Since this is kind of hydrolysis, so you are using water to break down N-acetyl glutamate into glutamate. Hence acetate is given out. Hence water is used. Hence the name of the enzyme is deacylase. Right? Done? Done. So this N-acetyl glutamate is related to the regulation of urea cycle. As I was talking about, that N-acetyl glutamate is one of the positive effectors of this enzyme CPS1, right? So we have something called coarse regulation and fine regulation of the urea cycle. Any cycle, any metabolic process in our body is obviously regulated in physiological conditions. Urea cycle obviously in physiological conditions would also be regulated. There is no exception to that. So regulation means that you can increase the frequency of this cycle, or you can decrease the frequency of this cycle. That is what regulation means, that you can fine tune or you can coarse tune this urea cycle. Now for fine tuning, why am I talking about fine tuning and coarse tuning? Listen to me carefully. N-acetyl glutamate is a compound which is affecting one step of urea cycle. So by altering N-acetyl glutamate, we can control urea cycle, but not on a great level, not on a very coarse or broad level. It's a fine regulation. Coarse regulation will come into play when we are talking about physical processes like starvation or maybe increased catabolism of proteins. That is the long-term coarse regulation of urea cycle. We will be talking about that. But since we are talking about N-acetyl glutamate, so I am explaining the fine regulation of urea cycle. So N-acetyl glutamate, since this is a positive effector of this step, so this will positively or rather stimulate this step to happen actually, right? Now, thing is, N-acetyl glutamate itself is being formed from glutamate. So if glutamate is high, N-acetyl glutamate will also be much more formed, and hence more urea cycle frequency, because N-acetyl glutamate is positively influencing the cycle, right? All right. Now there is another thing. This N-acetyl glutamate synthetase enzyme, this is also an enzyme, so this also has a positive effector. Who does the positive affecting work of N-acetyl synthetase? Means who positively stimulates this enzyme? That is, remember, arginine. This knowledge is not far-fetched, all right? This might seem like I'm stretching it, and what is the need of going to such details as to why energy is being formed and what is the effector of the enzyme which is forming energy, which is in turn a positive effector of a step of urea cycle. This is not far-fetched. Why am I telling this? This will be clearer when we will be talking about the disorders of urea cycle. These enzymes are very important, very crucial, because deficiency of these enzymes would lead to a variety of conditions called hyperammonemia. All right, we will be talking about that in just a minute. So I think the first step we can, that is what is kind of happening here. Now we come on to the second step, where what is happening actually? Carbamoyl phosphate is actually reacting with ornithine in the presence of ornithine transcarbamylase to form citrulline, right? And this is happening in the mitochondria. Citrulline is coming back to the cytosol, and ornithine is going into the mitochondria from cytosol. So there is something called ornithine, very important. Listen to me. Ornithine citrulline transporter. Ornithine citrulline transporter. All right, what is that? It is as the name suggests. This transporter is actually helping in the migration of ornithine from cytosol to mitochondria. All right, and is also helping in the migration of citrulline from mitochondria to cytosol. All right, so this transporter is again very important, because again any defect in this transporter will lead to disorders. We will be talking about that. Now the next step was the formation of Argino succinate from citrulline itself by the enzyme Argino succinate synthetase. Obviously 1 ATP 2 phosphate bonds were broken in this step, and Argino succinate was formed. All right, now just you may remember this. I don't feel that it is very important, but this thing, the PPI here, the 2 phosphates which are actually liberated, they act as inhibitor of this enzyme Argino succinate synthetase. This is something that I actually read from the book, and so I kind of mentioned it. I did not find the impetus to actually kind of, remember this or anything like that, but you may remember this, right? The next step is formation of arginine. That is from arginosuccinate. Arginosuccinate lyase enzyme breaks down arginosuccinate into arginine and fumarate. Fumarate is coupled with Krebs cycle in mitochondria, and arginine goes on with the urea cycle. So this step is very important because this is the splitting of the bicycle. We were talking about the urea bicycle, right? So this is the part where the cycle splits and the bicycle is formed. All right. Now, here is the thing. The fumarate, which is formed, is actually an inhibitor of this enzyme, arginosuccinate lyase. This is very interesting. This is interesting, and this is worth remembering, all right, because this is interesting. This fumarate is an inhibitor of arginosuccinate lyase. So this should be a problem because this is product inhibition which is happening here. You're forming something, and that something itself is inhibiting your step. That is not something which we want, or the body wants, or the cell wants, right? So what a beautiful game nature has played here is that the fumarate does not pose a threat to arginosuccinate lyase, although it is a potent inhibitor of this enzyme. Why? Because they are separated. They are compartmentalized. What does that mean? You see, fumarate, after being formed, rushes into mitochondria. Because, as I said, this enzyme fumarase is mitochondrial, right? So since this is mitochondrial, so fumarate rushes to mitochondria, and fumarate does not inhibit the enzyme arginosuccinate lyase, which is actually in the cytosol. So due to different places, due to their different sites of actions, arginosuccinate lyase is potentially saved from fumarate's inhibitory action. All right. So this is something interesting. Hence, arginine is formed, and the next step is the last step, which is the regeneration of ornithine and urea being formed due to H2O, one molecule of water entering into this and reacting with arginine to form one molecule of urea and ornithine. Ornithine is again used back in the cycle. All right. So ornithine is being regenerated and it is not changed, right? Ornithine itself is not getting changed into anything. It is getting changed into many compounds, but after all it is being regenerated. So that's why ornithine is said to be the catalyst. It is the kind of the catalyst of urea cycle, because catalysts themselves by definition are unaltered after chemical reaction. So that is what ornithine is also following. So that is also an important thing which we may consider. Now let's get on with a very short thing, which is the ATP part. Two ATPs, so two phosphate bonds broken. One ATP but two phosphate bonds broken. So 2 plus 2, 4 phosphate bonds are broken. 4 phosphate bonds are broken. So the net expenditure should be 4 high energy phosphate bonds as far as the energetics of urea cycle is concerned. However, there is a twist to this story. The plot twist comes with this part. You may remember that in Krebs cycle, we had formed NADH, right? NADH was formed. NADH was formed in the step malate to oxaloacetate, right? So from malate to oxaloacetate, NADH was formed. NADH, upon going through the electron transport chain, would yield 2.5 ATPs. 2.5 ATP. So that 2.5 ATP is gained from the urea bicycle, right? So from the urea bicycle, that 2.5 ATP is gained here, right? So we have to deduct that. And what are we left with? 1.5 ATP expended. Alright, 1.5 ATP is used in this urea cycle. So that is something that we need to actually consider. That is 1.5 ATP is needed for the entire urea cycle. Now we were talking, I talked about the fine regulation pertaining to N-acetyl glutamate. So let me talk about the coarse regulation as well. Alright. Now you see, as I was talking about, the different physical processes actually stimulate the coarse regulation of urea cycle. Coarse regulation means the broad regulation, the long-term regulation of urea cycle. That is happening when a person is starving. Suppose he is starving, or there is increased catabolism of proteins. So in that case, what happens is urea cycle needs to be operated at a very higher level, because you see, when starvation is occurring or when there is catabolism of proteins, the protein source in our body is getting depleted very fast, right? Amino acids are metabolized very fast, so the ammonia is also being formed at a very high rate, and its disposal in the form of urea also has to be at a very high rate to keep up. So urea cycle also needs to operate at a very high level. Now what happens is, for this urea cycle to occur fast, these enzymes, they are actually synthesized in an increased manner. But how can these enzymes be actually synthesized fast or in increased amounts? That is only possible if we alter genetic level. If the alteration is in the genetic level, then these enzymes are synthesized quite fast and quite, I would say, in increased amounts. So that would lead to more frequency of the urea cycle, because there is more amount of enzyme there, right? So that I think does justice to the coarse regulation part of the urea cycle. And the fine regulation I already talked about, that the N-acetyl glutamate which is being formed, it is a positive effector of Cps1, which is the rate limiting step actually. So it is very important to remember this molecule N-acetyl glutamate, and this itself is formed by glutamate plus acetate. So glutamate plus acetyl CoA. Alright, so acetyl CoA is needed, and the enzyme which is actually needed is NAG synthetase, right? And acetyl CoA gives on coenzyme part A is removed and acetate is used up. NAG, after being formed, it may be broken down into glutamate by the enzyme deacylase, and here a water is used, right? This NAG synthetase itself is positively regulated by arginine. This is an important information which we will use just in a few minutes time. So I guess as a process the urea cycle has been dealt with. Now we come on to the very, very important thing, which is the reason why we are actually studying this process, is that the disorders which pertain to urea cycle. I'll be very fast. The upper hand that we have in this case is that the disorders of urea cycle, they are not very varied, they are not very diverse, they are kind of the same thing. All right, now listen to me very carefully. If there is a kind of problem with any of the enzymes of urea cycle, right? So if any enzyme, suppose there is deficiency of an enzyme of urea cycle, then what would happen? That enzyme was obviously, it was catalyzing some step, right? So that step would not take place. That step would not take place. So if A to B was happening due to the enzyme X, and now this X has decreased in amount, or rather there is a deficiency of this X enzyme, then this step would not take place. So B would not be formed or formed in very less amounts, and A would kind of accumulate, because A was supposed coming from Y, and this step is all right. J is the enzyme in this step. Y to A, this step is fine. There is no problem with this step. The problem is with A to B, right? So this step would be kind of a stoppage. So A would accumulate. This is the problem. This is the main theme of the disorders of urea cycle, or any biochemical process. Any biochemical process, that is the thing. All right. There is accumulation of a particular thing, right? Now, let's talk about the different enzymes. So let's bring back that page, and let's give it a quick go. Cps1, carbamoyl phosphate synthetase 1, is the first enzyme which we have. Suppose there is a deficiency of Cps1. So if there is a deficiency of Cps1, right? So Cps1 deficiency is there. What would happen? This step would not take place. And what would accumulate? Carbon dioxide? No, obviously not, because carbon dioxide, the level of carbon dioxide is not dependent on urea cycle. Carbon dioxide is not related to urea cycle. The level of carbon dioxide is not related to urea cycle, but the level of ammonia is related to urea cycle. So ammonia would increase. And the increase in ammonia is kind of a common theme to almost all the disorders of urea cycle, because ammonia is the first thing. It is the starting thing. So anywhere you get a block, kind of there is a tendency of ammonia being formed. However, let me warn you about something. The earlier the enzyme gets deficient, or rather, let me put it in some other way. The earlier enzymes, if they are deficient, there is more danger. If the later enzymes are deficient, there is less danger. The reason is, if the earlier enzyme, suppose carbamoyl phosphate synthetase 1, this enzyme is deficient, then, or suppose OTC, this is OTC, ornithine transcarbamylase, this enzyme is deficient, then there is ammonia increasing tendency, hyperammonemia, and ammonia is very toxic. However, these intermediates, Argino, Succinate, Arginine, these are not that, I would say, toxic. All right. So these intermediates are not that toxic. So their increase in our body, they would lead to obviously deleterious effects, but that would not be as devastating as hyperammonemia due to the deficiency of the earlier enzymes. So this is a common theme that we should take into consideration. Now, if Cps1 is actually deficient, that is carbamoyl phosphate synthetase 1 deficiency, then there is obviously hyperammonemia. All right. Now, what kind of a deficiency, or rather what kind of a disease is this? This is actually an autosomal recessive disease. What does that mean? It means that this Cps1, carbamoyl phosphate synthetase 1, this is an enzyme. So obviously an enzyme is a protein. So for the formation of the protein, the different steps must have taken place. Transcription, translation of protein, that must have taken place. So the gene which is responsible for the formation of this protein, that is Cps1, there is a problem with that gene. Either it is defective or it was not inherited. And that gene is in the autosomal chromosome, not in the sex chromosome, or rather X chromosome. It is in the autosomal chromosome. And the condition is recessive, which means that only the Cps1 deficiency, we see this condition only when we get a defective copy of the gene both from mother and the father, the male and the female. That is why it is recessive. If it had been dominant, then only one faulty copy of that gene would have been enough to manifest the effects of this Cps1 deficiency. But it is recessive, so that means that that gene, or rather this condition, is only manifesting itself when defective copies are from both the sexes. So obviously that is not related to biochemistry, but that is related to the general knowledge of biology. So this is an autosomal recessive disorder, and as we will be seeing in the different enzymes deficiency, almost all the enzyme deficiencies are autosomal recessive except one. All right, except one. I'm telling which one. Let us go in an order. All right, that would be much faster and efficient also. Now, I had said that carbamoyl phosphate synthetase, there is a deficiency in that. Let's consider there is no deficiency in this enzyme. What is the next enzyme? Ornithine Transcarbamylase, OTC, Ornithine Transcarbamylase. So let's suppose that there is a deficiency of OTC. What would that do? So if there is a deficiency of ornithine transcarbamylase, then obviously there would be hyperammonemia. Obviously, hyperammonemia is obvious, right? Now I had forgotten to say something in this case. You see, when ammonia level is elevated in the body or blood, that is known as hyperammonemia. But hyperammonemia due to Cps1 deficiency is known as hyperammonemia type 1, and due to OTC deficiency is known as hyperammonemia, I'm abbreviating this, type 2. Alright, so the hyperammonemia which is actually due to Cps1 deficiency is hyperammonemia type 1, and that due to OTC deficiency, ornithine transcarbamylase deficiency, is the hyperammonemia type 2. Alright, so this is something that I wanted to say. Now let me cite another thing. Suppose there is increase in ammonia in both cases, right? So if there is an increase in ammonia in the body, what would the body try to do? The body would still try to actually follow all the steps of whatever it would do if it had been a normal case. Am I being clear? Suppose it had been a normal case and there is ammonia being formed from different parts of the body, then what would have been done by the body? I had said this in the previous class. Ammonia would be transported to the liver. That is what would happen. Can ammonia be transported to the liver as such? No. It is transported in a trapped form. What is a trapped form? The trapped form is, very good, glutamine. So glutamine is a trapped form of ammonia because glutamate actually traps ammonia in the presence of the enzyme glutamine synthetase, right? And glutamine is formed. Again, in the liver, we have this enzyme known as glutaminase, which would actually revert back the glutamine to glutamic acid and ammonia. Obviously, that would require a molecule of water. Now, the thing is, if ammonia is increased, body would still try to trap that ammonia. So glutamine level will also increase. This is something which is related to hyperammonemia of both things. That is, glutamine level would increase drastically. How are we able to say this? We have to measure this, obviously. So we need to extract something and measure the increased amounts of glutamine there. What is that extracted thing? Yes, it is CSF. And also blood. In the different biochemical, pathological investigations, body fluids which are actually used include CSF, blood, and there are many other things, obviously. There is urine, there is saliva, and also many other things. So in both these body fluids, there will be an increase in glutamine. Why? Because there is an increase in ammonia and the body is still trying to trap it. Now let me tell something about OTC deficiency as such. Listen to me very carefully. Let me again bring this back. If there is a decrease in OTC, ornithine transcarbamylase, there is a deficiency in this enzyme, then this step would not be taking place well enough. So what would happen? There would be an accumulation of carbamoyl phosphate, obviously. Where would that accumulation be? This is where? This is the mitochondria. So the accumulation would be in the mitochondria, right? So carbamoyl phosphate would accumulate in the mitochondria. This is the normal urea cycle. Give this a look and tell me, is carbamoyl phosphate in the cytosol anywhere here in the cytosol? No, it is not. It is in the mitochondria, because in the mitochondria itself it is forming citrulline and citrulline is continuing. So carbamoyl phosphate as such is not coming out of the mitochondria. But obviously that is only the physiological part, only normal physiological part that is happening. In cases like OTC deficiency, or what is the other name for this? Yes, hyperammonemia type 2. Carbamoyl phosphate, due to increased accumulation in mitochondria, would kind of seep into the cytosol. It would seep into the cytosol. And this has adverse effects. What adverse effects? If you have already studied nucleotide metabolism, you would be able to correlate what I am going to say now. If you have not, don't worry, just give me a patient hearing. The thing is, OTC deficiency causing carbamoyl phosphate to come out in the cytosol would lead to obviously a halt in the urea cycle. It would obviously lead to a halt in the urea cycle. But that is not what I am pertaining here. You see, carbamoyl phosphate is one of the, I would say, metabolites in pyrimidine synthesis. That is why I said nucleotide synthesis. If you have read that, you would understand this. So if carbamoyl phosphate comes into the cytosol, it is used in pyrimidine synthesis, right? It is incorporated in the pyrimidine ring. Now, in pyrimidine synthesis, obviously there are a list of steps. The regulatory step, the rate limiting step of pyrimidine synthesis is bypassed if carbamoyl phosphate, high levels of carbamoyl phosphate seep into the cytosol and are incorporated into the pyrimidine ring in case of OTC deficiency. I am repeating again. OTC deficiency is leading to carbamoyl phosphate accumulation, right? due to accumulation in the mitochondria, it is seeping into the cytosol. In the cytosol, this increased levels of carbamoyl phosphate is used in the pyrimidine synthesis, right? However, the step in which carbamoyl phosphate is used in pyrimidine synthesis is not the regulatory step, and by that time the regulatory step has been bypassed already. So that means that increased levels of carbamoyl phosphate would lead to a condition which is known as orotic aciduria. Orotic aciduria is the name of the condition which occurs due to this OTC deficiency and due to the reason which I said before this. So that's that. Alright, so please remember that. Now we come on to the next enzyme, which is okay. Now, before going on to the next enzyme, let me tell you about something. Do you remember that I had talked about a transporter here? The Ornithine Citrulline Transporter. The Ornithine Transporter. If there is a problem with this transporter, then ornithine would be trapped in the cytosol. Alright? It would be trapped in the cytosol, and there would be no way for ornithine to go into the mitochondria because its transporter is defective. Right? This transporter is defective, so ornithine cannot go into the mitochondria. So ornithine would build up. That is known as hyperornithinemia. Hyperornithinemia. Due to increased levels of ornithine, due to ornithine not going to this mitochondria, so urea cycle is suspended. If urea cycle is suspended, hyperammonemia is also taking place. Right? Hyperammonemia is also taking place. Right? So hyperornithinemia, hyperammonemia, and due to the defective transporter, another thing takes place which is known as Citrulline Urea. Alright, let me explain it. Very fast I will explain this. So what's actually happening is, I talked about the ornithine transporter. There is some problem with this ornithine transporter. So if there is a problem with the ornithine transporter, what would happen is ornithine level would go on. So that is known as hyperornithinemia. That is the first thing. Second thing is the ammonia levels would also go on because urea cycle is suspended. So hyperammonemia. And another thing would take place. Now give me a patient hearing again. You see, since ornithine is not being able to go on to mitochondria, what the cell, or rather what would be attempted by the cell, is to bypass this ornithine part. How it would try to do that is, lysine, the amino acid lysine, is carbamoylated to form homocitrulline. So what should have happened is, ornithine should have been carbamoylated to form citrulline by carbamoyl phosphate. But because ornithine is not present, so the body tries to make an adjustment, an improvisation if you mind. So that improvisation is, the lysine amino acid is carbamoylated to form homocitrulline. So homocitrulline is also increased, and that is known as homocitrulline urea. Alright, homocitrulline urea. Okay, it's kind of a tongue twister, so I'm actually having a bit of a problem. So homocitrulline Citrulline Urea. These are the three things which happen if there is a defective ornithine transporter or deficiency in ornithine transporter. Alright. So these three H together, they are known as the HHH syndrome. So these are not random things, these are not random effects. These actually pertain to a very important thing, which is HHH syndrome. So the HHH syndrome is coming from deficiency in ornithine transporter. Right? Right. So that's that. Now again, I'm very sorry, but I forgot to mention a very important point. You see, this ornithine transcarbamylase deficiency, this deficiency, or rather this disorder, is the only X-linked disorder of urea cycle. It is the only ornithine transcarbamylase. Do you remember? You know, hyperammonemia type 2 ornithine transcarbamylase deficiency, which we had talked about just a few moments ago. That deficiency, that disorder is X-linked. I had said that most of the disorders are autosomal recessive. In urea cycle disorders, most of them are autosomal recessive. But there is one exception, I had said that, and that is ornithine transcarbamylase deficiency, which is X-linked. Right? So please remember this. Now, we have moved on to the next enzyme, which is Argino Succinate. So this is the product which is being formed. So Argino Succinate Synthetase. So if there is a deficiency in this enzyme, then obviously Argino Succinate would not be being formed, and there is Citrulline Urea, or rather it's actually known as Citrulline Urea itself, and also it is known as citrullinemia. So Citrulline Urea means that the citrulline is, due to its elevated levels, it is being passed in the urine. So that is why it's Citrulline Urea. And citrullinemia means overall elevated levels of the citrulline. That is due to AS synthetase deficiency. Alright. And obviously there will be an hyperammonemia. But as we are going farther away from the earlier steps, the threat is decreasing. Because as I said, the intermediates are, yes, they are less toxic. Alright. The next enzyme that we have is arginosuccinate lyase. If there is a deficiency of arginosuccinate lyase, then we have this condition in where arginosuccinate would increase in our blood. So there is arginosuccinic aciduria. Alright, arginosuccinic aciduria. Now there is another important point related to arginosuccinic aciduria. This arginosuccinic aciduria means that arginosuccinic acid or arginosuccinate is increasing. That level is increasing. Why am I talking about this by writing about this is because this arginosuccinic aciduria has a very characteristic manifestation as far as physical manifestation is concerned. That manifestation is what we say to be trichorhexis nodosa. Trichorhexis nodosa. I feel like that is the correct pronunciation. I hope so. Trichorhexis nodosa. It means it has something to do with hair, isn't it? trichorhexis. The trichorhexis, so here it's related to hair. So trichorhexis nodosa, that is related to hair, a hair condition. Nodosa means node-like formation is actually happening. So this is very interesting to note that a disorder in urea cycle is getting its manifestations at the physical level in the hair, that is node-like formation. So I think that is very interesting and hence worth remembering. So how is trichorhexis nodosa manifested? Due to what? Argino-succinic aciduria. Why is argino-succinic aciduria taking place at the first place? Very simple. Argino-succinate is being accumulated. So which enzyme is leading to the accumulation of arginosuccinate? That is arginosuccinate synthetase. Right? Am I right? Arginosuccinate synthetase enzyme, that's deficiency is leading to trichorhexis nodosa? No. I was actually kind of playing, so just to see. So the thing is, it is the lyase deficiency. Arginosuccinate is accumulating. That means that the reaction which should have taken place with arginosuccinate is not taking place. What reaction should have taken place with arginosuccinate? Arginosuccinate should have lysed into arginine and fumarate. So this enzyme, arginosuccinate lyase deficiency, is manifested by arginosuccinic aciduria, which is manifested by trichorhexis nodosa. Alright. Then we come to the next enzyme, which is arginase. And arginase deficiency, obviously it is very mild because we have moved on to the last step. So the metabolic intermediate is also less toxic, the least toxic in fact. And arginase deficiency, if I have to say something, it's nothing very special. There will obviously be an accumulation of arginine in the body, and also there will be hyperargininemia and argininuria. So hyperargininemia means arginine is getting accumulated, and argininuria means arginine is being excreted also through the urine in higher levels. So that is that. So with this, we kind of complete the disorders of the urea cycle. Now the thing is, just one extra point I would like to add in general. It is that in the disorders of urea cycle, we have some general characteristics, like general manifestations are seen. It might be specifically a enzyme deficiency, and hence that substrate would be accumulating, and hence the symptoms are shown, right? But there are some general symptoms which are actually pertaining to hyperammonemia. What are they? They are what we say to be encephalopathy. So apart from encephalopathy, other clinical manifestations of, generally speaking, of disorders of urea cycle include obviously hyperammonemia and, very important, that is respiratory alkalosis. What is this respiratory alkalosis? Very carefully listen to me. So what happens is that due to increased ammonia, which acts as a stimulant, the body reacts by hyperventilating. Hyperventilation starts. Due to hyperventilation, there is very fast, removal of carbon dioxide from the lungs, because ventilation is going on. So carbon dioxide is released. Carbon dioxide is actually removed very fast from the lung alveoli. So carbon dioxide is very closely related to the acid-base balance in our body, right? So if it is removed very fast, then to cope up with the situation, what happens is H plus and bicarbonate react in the presence of the enzyme carbonic anhydrase to give H2CO3, which would in turn dissociate into H2O plus CO2. So this would actually cope up with the increased hyperventilation and with the increased removal of carbon dioxide from the lungs. So this is the way in which the body reacts. So in this way we can say that H plus protons are being what? Removed from the body, released from the body, removed from the body, because carbon dioxide has to be formed. So there is alkalosis in the body, and that is due to respiration. So respiratory alkalosis due to what? Disorders of urea cycle, that is hyperammonemia. So these are some general features which are actually taking place. Also, this ammonia, I had already said that it is very toxic, and if it is accumulating, then mostly the part which is damaged mostly is the nervous system, the central nervous system. The reason is that brain is very sensitive to ammonia. Our brain is very, very sensitive to ammonia. And in fact, as I had said, that the glutamine levels would increase. It turns out that the largest source of this glutamate is the brain itself. So glutamine increase is also related to brain being affected by hyperammonemia. Alright, so if we have to treat hyperammonemia, then we have to obviously eat very less protein in our diet, obviously, and milk also has to be avoided. Why? Because milk is a source of citrulline. And if citrulline is present in milk, and if we are consuming it, then we are adding citrulline to our body, which is not good, because citrulline is what? A part of the urea cycle. So it would lead to accumulation of more metabolites of the urea cycle. So we have to avoid that. So although there are some other things also, like hyperammonemia in details, we could have talked about that, but I feel like for now this would suffice. So I think we have completed with urea cycle. We have done it quite a bit of justice. Although hyperammonemia, as I said, it would require a bit more discussion. Anyways, so I feel we have done well today. Very well. Okay. Bye. Goodbye. Peace.